Whenever you're given a question about a medication or a drug that's being given in different amounts like this one here, you'll always want to test a group that doesn't receive the drug, and that's called the control group. So that's what we need here. We've got two groups taking this medication. We want to add a third group that doesn't take the medication, because that's the only way to find out if the drug is effective, is by comparing it to the group that didn't receive the drug. So that's how we could remove the error in the setup here, test a third group that doesn't receive a medication. So when it comes to a study involving drugs, always make sure you have a control group that doesn't receive the drug. Okay, let's keep going here. What would improve a weakness in this sampling technique? Well, looking at the graph, we're missing a lot of data points here, and that is a weakness in the sampling technique. So what's going on? After a nuclear power plant explodes, that's crazy, scientists measure the radiation intensity at far and close distances to the site. And that makes sense, because at farther distances here, if we come straight across, the radiation's pretty low, but at closer distances, the radiation's higher. The problem is, again, we don't know what's happening in between. But to solve that, we just need to collect more data. Then we'll find out if the data points fall in a line like this, or if they drop off quickly and then flatten out. So whichever answer mentions collecting more data, that's the one that we want here. And the first one, it does that. Samples should be taken across a range of radiation levels. In other words, we'll find out about these radiation levels as long as we take measurements at all of these different distances. That'll fill in all of the gaps here. So anytime your graph is missing information, just go ahead and collect more data, and that'll strengthen the weaknesses in your sampling technique there. Which change would improve the experimental design? 3 lakes near an old mining site are at risk for contamination. Scientists collect 1 fish from each lake, let's highlight that, to test for the presence of heavy metals, such as lead and mercury. The fish are dissected, and tissues known to accumulate heavy metals are analyzed. So again, what would improve the design here? Well, we're looking at 3 lakes to see if they're contaminated, but we're only looking at 1 fish from each lake. But you can't draw a good conclusion from just 1 sample. So to improve, we want to collect many more than 1 fish. And see, it has that. By testing more than 1 fish from each lake, that definitely improves the design, so that's what we want. Because if you only test 1 fish, it might just have a low lead and mercury level, while many of the other fish actually have higher levels. So if you grab 30 samples instead of 1, then you can get an average of both of these and draw much better conclusions. In other words, always grab a sample size greater than 1, and that'll automatically improve the design of your experiment here. What else should the researchers measure to test their hypothesis? A group of researchers is investigating the effect of commercial airplanes on air quality. Researchers hypothesize that pollutants, such as carbon monoxide, would appear in higher concentrations in an area with high volume air traffic. They collect air quality samples in one high volume area at a common flying altitude. So again, what else should these researchers measure? Well, what are they measuring to begin with? They're measuring this pollutant, carbon monoxide, to see if it's in a higher concentration within a high volume air traffic area. But the problem is, we don't know if something is in a higher concentration until we know what is the baseline or normal level of it somewhere else. So which answer is going to measure that? The first one actually does that. We want to measure the concentration of carbon monoxide in a low volume air traffic area. That'll give us an idea of what's normal, so we could see if this one's higher. In other words, if this one we measure to be 2 parts per million carbon monoxide, but this one we measure 35 parts per million, it's only now we can see, oh yeah, this is much higher because we know the normal level of it. So that's it. You'll always want to know a typical or baseline level to draw the conclusion that it's much higher somewhere else. Here's another video with GED Science Experiments and Investigations. Check out my website for practice problems just like these and others as well. Let me know what questions you have, what else you want me to cover. Good luck, you got these. We'll see you in the next video. Toodles.